So, you guys know Homer, he was a Greek poet. Okay. So, he wrote two poems which are very important in the literature whatever he wrote. One was Iliad and another was the Odyssey. Yeah. So, it is equivalent to that, right. So, Odyssey is the story of the king Odysseus, right, when he fought the war of Troy, and when he was coming back to his home after 12 years with some of his ministers who were left over, they had to fight with this monster who had one eye, that's called as Polyphemus. So, now there was some cartoon character also once comes, I think, which has only one eye or something, I do not know. Yeah, some cartoon character also comes. But if you think about all these things, no, we can think that, oh, maybe it was his imagination, and he must have just thought something like that. You want to create some villainous character, so how bad you can make it, so you put some one eye and all that stuff, right. But in 1950s, in this particular state of Idaho, farmers, they started, it happened in United States, and farmers started seeing these kind of sheep in their farms. So, this is and then this is called as cyclopia or cyclopic lamb. It is not only in the sheep, it is present some of I do not want to put the picture, but there are some human faces also which are similar. So, they called it monster sheep. Now, the question is why? How can you have these kinds? So, these farmers were really worried by Vaishnav, because you do farming, sheep are very important, and you find something like this, what do we do, right, Sayan? Then these people, what they did? They went to the, of course, they went to the scientists, of course, they have to come to us, right. So, they went to some scientists in the agriculture department, and those people started doing some research, and they realized that the mother sheep, when this baby sheep was in, the mother sheep when she was pregnant, that mother sheep grazed upon this plant called corn lily. This corn lily is a very common plant which is found in that region. And eventually people found out that this corn lily has a compound called cyclopamine, which goes and inhibits your Hedgehog signaling pathway, and that is how the whole Hedgehog signaling pathway was discovered, and cyclopamine, which is the inhibitor of Hedgehog signaling pathway, was discovered. And then people showed eventually that if you inhibit the Hedgehog signaling pathway, that is what I was telling you, for your craniofacial development, digit patterning. In adults, I will come to that slide. In adults, is very important during the development, and that is how this pathway was discovered. Oh, that I do not remember, but it happened in early 1960s or sometime. Phil Bichie was the person, he did that, and then there have been enormous work which has been done by so many people after that. I mean, I enjoy talking about it because it involves primary cilia. So, I will show you this video. So, this is the tiny hair like structure. If you go to my web page, you will find this video anyways. So, this tiny hair like projection, what you see, that is the primary cilium, and the reason why I am showing you this is not to promote my lab, is to show that in case of mammals, the Hedgehog signaling pathway takes place through this tiny organelle. It is a hair like structure around 3 to 5 micron, 250 nanometer diameter, and you see that, you can see that there is something which came in, some GPCR kind of structure just went inside. Now, there is some machinery, I will replay the video. Now, there is some machinery which we take it inside, then there is additional machinery which is coming, and that is trying to take it out of the cell, right. So, these primary cilia, they host all the G protein coupled receptors, or the receptor molecules, secondary signaling molecules inside the structure, and most of these molecules will be taken inside the cilium by the machinery which is called as intraphaginal transport machinery. And then you see these green color structures, those are tubulin tracts. So, cilium is made up of tubulin, and this tubulin which is in 9+2 arrangement, 9+0 arrangement in primary cilia, that is kind of a cylinder, and these are motors. The orange color thing what you see is your kinesin motor, and this kinesin will walk onto the cilium, and that is how it is taking. So, this GPCR kind of structure what you see, it is one example, like all your Hedgehog pathway receptors which are important for the signaling. They are carried like this inside the cilium, they function over there, whatever they are supposed to do, they do it, and then once they do go to the cilium, and this is something which I try to show, that how we discovered the pathway, how these receptors will be recognized and removed out of the cilium. But then there is a machinery which will remove. So, once the receptor is going inside, it is going with the kinesin motor, and now you can see that orange color changes to pink color or magenta color, which is because the motor changes. Now, it is a dynein motor which is trying to take the receptor out. The reason to show this is because in case of mammals, vertebrates especially, primary cilium is very important to carry out the Hedgehog signaling pathway. As I mentioned, there are 3 Hedgehog ligands, IHH, DHH and SHH. IHH is Indian hedgehog, DHH is desert hedgehog, and SHH is sonic hedgehog. These 3 different hedgehog ligands are there. They of course get secreted out like what we saw in case of wind, and then there are specific receptors for this pathway. We will talk about the pathway in detail in next couple of slides. So, it is very interesting during your development, this is what is shown over here is the development of mice and how the hedgehog is very important, and if you pay little bit careful attention, you can see that the left right axis, what do I mean left right axis is nothing but your heart is on left side, and this organ patterning which happens now is very interesting. So, during your during your development, so at one point there is one cilium which is present, and that keeps moving like this in circular pattern, that is called as the nodal cilia, and this cilium creates the gradient of calcium or something which is very important. If you remove this, you must have heard some cases where they have situs inverters. So, instead of having your heart on left side, it goes to the right side and your whole organs change. That happens because this nodal cilia is not functional, and that is one of the characteristic of some of the ciliopathies. And most of those ciliopathies are nothing but the disorders which are caused because of there is defect in the cilium, whether it is motile or primary, it is a different thing. If it is non-motile cilia, which is mostly through the Hedgehog signaling pathway. Now, if you pay a little bit careful attention, this is E is nothing but it is E embryonic stage, right, if it is 8.5, 9.5 and all that. The left right patterning, limb development, cerebellar growth. So, there have been examples where people will see microcephaly, hydrocephaly. You guys know what is hydrocephaly? Like you will say very big head, and that is because of the cerebrospinal fluid will get accumulated, and microcephaly means your development will not happen and they will have incomplete development. So, all these things happens with associated with the ciliopathy phenotypes. Neural tube patterning. So, when you are developing, during your development, there is something called as neural tube which is being, so there is a the packs and there is a gradient of different molecules which happens, and this whole thing is regulated by your Hedgehog signaling. If it is not proper, the closure of neural tube will not happen, and that is being regulated by Hedgehog signaling. So, sonic hedgehog, Indian hedgehog and desert hedgehog, all these 3 guys, they play important roles at different stages. Like SHH, which is important for left right patterning, limb development or neural tube patterning. IHH, Indian hedgehog, is important for your bone growth and pancreatic. Many of these patients, they have, apart from having craniofacial developmental problems, they do have short ribs and they have other issues because of their bone development or other development will be affected. DHH is very important for your gonad development. So, DHH and IHH together, they play very important role whether it is male or female, and that is very important. So, in fact, in females, if you during the development, so in females for the androgen synthesis as well, Hedgehog signaling is very important. If you look, so there are certain factors like oocyte development during the oocyte development and follicle development. Hedgehog signaling in adults, I am talking, Hedgehog signaling play important role, like GDF9 is one of the factor. So there are two types of cells, one is theca cells and another is granulosa cells. These cell types, so granulosa cells will secrete Hedgehog, which goes and activates theca cells. GDF9, which is coming from your oocyte, will go and induce this, and then it will go and activate the pathway. It will secrete the molecules, then it goes and activate the in theca cells, and that's how the development happens. We are not going to talk about it. I am just giving you an example how important the Hedgehog signaling pathway becomes based on the organism we are talking about, and in combination of sonic hedgehog and Indian hedgehog, it is important for gastrointestinal development as well as prostate development, and it depends, it varies based on the stage of your development. During very early time, when your neural tube patterning happening, that's when your sonic hedgehog becomes very important. And remember, as I said, in vertebrates, everything happens through primary cilia. No primary cilia, no Hedgehog signaling. In Drosophila, they don't have primary cilia, but still Hedgehog signaling happens, where all the components of Hedgehog signaling will be present on the cell membrane, not on the cilium. Is it clear? Good. So as we had talked about it, you guys remember in case of Wnt, you remember, Shine, in case of Wnt, the ligand gets secreted, and we saw there are multiple pathways how it gets modified, right, and we know any molecule which goes through secretory pathway, Vaishnav, it will go first to ER, from ER goes to Golgi, and then it will be secreted out. Similarly, where is olfactory? Oh, you are asking in general olfaction development. So the Hedgehog signaling, so there are certain reports which are very recent, which say that even in your ear, there is kinocilia, which is important. So in your hairs, if you look at it, there is a very nice pattern. They go like this. So they go. There is the development of cells, something like that, and there is one cilium. So I don't remember so clearly, very clearly, but these kind of those kinocilia or something which are present in your ears as well, so they also play important. And in case of olfaction, what I know is that I do not know about Hedgehog signaling, but what we know is that there are some GPCRs which are present in some of the cilia. Those cells also do have cilia in them, and they carry certain GPCRs, and which are very important too, because they are connected to the neurons. So it's similar like what I was talking about your retina and photoreceptors. They also carry certain primary cilia, but mostly depends on the GPCR signaling. But I don't recollect anything about Hedgehog signaling being important for the development of olfactory neurons or those. Oh, there I think you can, so one eye development, I think it goes with the cranial neural crest patterning, your neural tube patterning, and limb development. They miss some of this. So craniofacial development, all those defects will come. So that is one example, that's extreme example. I mean, there have been some examples where we have seen, so I was giving you example of cleft palate, right. So the ciliopathies are very interesting. Some of the kids, very small, there are certain ciliopathies where younger kids, they will lose the ability to hear, and they will be obese as well. So based on these combinations, you categorize them which kind of phenotype they would have. So in case of Hedgehog phenotype, you can have anything. It's not only based on one phenotype you assign that this is the problem. You need to have primary and secondary phenotypes, and then combination will tell you what is what. In case of medical, if you go and talk to a clinician, the clinicians will have their own way of making sure that this patient is because of there is a defect in Hedgehog signaling or not. But in general, these are the things which are very important. Hedgehog signaling is important for the development of all of them.
I said this like signaling is only important in developmental or other in the adults also.
Very good. So, in case of whatever I explained you earlier, that is very important for doing the development. In case of adults, which I was telling you about, is during the androgen synthesis, during the follicle synthesis. In case of females, especially in maintenance of reproductive organs, that is very important to have them. And there are certain cells, even in adults, the Hedgehog signaling takes place, but that is mostly for the tissue homeostasis, maintenance of the tissues. And people have seen that this Hedgehog signaling can go wrong in some of the glioblastomas. So, glioblastomas are the cancers which are Hedgehog dependent, where there is hyperactivation of Hedgehog signaling. And there are certain cancers where, so there is a combination of it, meaning in some cancers primary cilia will be lost and then the pathway gets hyperactivated. In some cancers, without, they are not dependent on primary cilia, but Hedgehog signaling gets hyperactivated because the glia transcription factors gets mutated and they are constitutively active. So, especially glioblastomas or the cancers associated to brain, there they have observed that there is hyperactivation of Hedgehog signaling.
So, the dysfunction of the signaling is due to the external stimulus or
Mutations can also be, we'll see some of the examples like that. So, for example, as we talked about Hedgehog ligand, that also gets processed, the Wnt ligand which gets modified and processed, Hedgehog ligand also gets. So, it will be synthesized as Hedgehog precursor, which has this N terminus and C terminus, and these are two parts to it, which is 20 kilodalton and 25 kilodalton, but there is one cleavage site at which that will get cleaved, right. And then what happens is, so they have given how does this cleavage happen in this reaction, then there is formation of this thioester bond because of the cysteine which is present over there, and the N terminal half of the molecule which gets cholesterol. So, cholesterol is very, very important in Hedgehog signaling because your ligand, Hedgehog ligand itself gets cholesterol. Cholesterol is added onto this Hedgehog ligand, and then at the N terminus, this side it gets palmitoylated, and the C terminus of this 20 kilodalton part teja. So, you have 400 kilodalton protein or 470, 20 kilodalton, and then remaining molecule half. So, this 20 kilodalton whatever you have tiny space molecule, that gets two modifications, one is cholesterol addition and another is palmitoylation. Is it clear? And this gets secreted eventually, and then there is a gradient of this Hedgehog signaling which happens. So, now we will zoom into the pathway, how does it happen. Again, what I am explaining is what happens in vertebrates, that is why I have put a primary cilium. Okay. So, we will be talking about in that context. Yes. It will happen in your ER and then at the Golgi. ERs do some of the smooth ER especially do the lipid, they are very important for the lipid biogenesis and all, and cholesterol, I think they do, some of the ER do have some cholesterol into them, right. So, these cholesterol modifications will happen in ER. Good. So, now, so whatever I have shown over here, it is extremely complicated and there are 1000 names which appear over there. We will go one by one. Okay. So, going back to Hedgehog signaling pathway. So, what I have shown here is how the processing happens. There are too many molecules. One molecule which everybody should always remember when we talk about Hedgehog signaling is glia transcription factors, which is equivalent to your beta catenin in Hedgehog signaling. So, what happens over here, and this is very interesting because all these signaling pathway do involve your cyclic AMP. In case of your beta catenin, we did not talk about it, but these guys, there is role of cyclic AMP as well, and I will tell you how. So, in case here, so there are three receptors which are very important in case of Hedgehog signaling. One is patched, right. This is actually the Hedgehog receptor. Then there is smoothened, hope my spelling is correct. This is the pathway transducer. And then there is GPR161. This is another GPCR. For long time this was supposed to be an orphan GPCR, meaning उसका कोई ligand नहीं है, agonist नहीं है, antagonist नहीं है. For long time. Very recently there was a paper in NSMB, Nature Structure of Molecular Biology, where they resolved the structure of this GPCR. Brian Kobelka's postdoc, Ashish Manglik, who is at the UCSF, who dissolved the structure. He is the same person who dissolved the structure of Smoothen as well. Cryo-EM is everywhere, so people are doing left and right. So they developed certain methods where they use nano discs, and then because these are membrane proteins, they are not easy to make the structures of. So you use nanobody based or nano discs based purifications, and then you can make the structures. That they use cryo-EM, and then they did it. So remember, patched, there is patched one and patched, yeah. The difficult part is, in case of membrane proteins, one of the challenge people mostly face is to express and purify the protein, because you need tons of that. First of all, like you need more protein. Expression is not a problem, but purification becomes a problem. Huh. Oh, no. In case of orphan receptor, people did not know what is the ligand for it. They did not know. They did not know. I do not know why. The question is why they did not know. Because I don't remember why they did not know. No, there was some literature, but I never. I worked with this receptor, but I don't recall it. Huh. Yeah, so for example, I'll give you, when, if you remember, once I told you, let's say there are 600 GPCRs which are known, right? Out of these 600, I do not know the exact number, but I gave some number to you guys. 800. Out of that, over 400 or over 500 receptors are, they are they are sensory. Sensory meaning they will sense the olfactory receptors, your photoreceptors and all that, and there are another 300-350 molecules which are non-sensory GPCRs, where they can get activated or inactivated by a physical ligand. In this case, for example, you know that there is a hedgehog which is the ligand which goes and binds to certain receptor. So in that case, you are patched. It doesn't become orphan, because we know that there is hedgehog which goes and binds to this. Like that, so there was a Cell paper which was published by Saikat Mukhopadhyay and Peter Jackson at Stanford long ago in 2017 or 2018. They showed they are the first people who actually discovered GPR161 as important hedgehog pathway molecule. But that time they did not know that if there is a particular ligand that goes and activates this receptor, that's why that was called as, this is very recently discovered GPR161. That's why people thought, they did not know whether it is, that's why they called it as an orphan receptor. However, if you remove this receptor, hedgehog pathway gets hyperactivated, which means that it is a negative regulator of the receptor. That is what they knew. Okay? And whenever you activate hedgehog pathway, this receptor disappears, this function does not happen. Make it more complicated, bringing in your cyclic AMP, what happens in hedgehog signaling pathway is that your GPR161 is G alpha S coupled receptor. Now, do you remember, Rutuja, G alpha S meaning what will happen? it will activate. So, G alpha S coupled GPR161 will activate adenyl cyclase. There is more cyclic AMP. This happens when Hedgehog signaling pathway is shut. When pathway is inactive, when Hedgehog signaling pathway is off, that time there is more cyclic AMP. And what we are talking about, they are talking about the cyclic AMP which is present in this teeny tiny space of primary cilium, but it does play very important role in passing the information to the downstream targets. And what is the downstream target? Is the processing of your Gli transcription factor, because Gli transcription factors are the ones which go and bind to the promoter, leading to the regulation of Hedgehog pathway genes. Is it clear? We will come to that part. However, smoothen, which is another GPCR, this guy is G alpha I coupled. What does it mean? It will inhibit, which means that in case of Hedgehog signaling pathway, whenever pathway is activated, there is less cyclic AMP. So, it is acting opposite way. We always think that pathway active means more cyclic AMP. In this case, pathway active means there is less cyclic AMP. Is it clear? Good. So, patched, you see that patched, which is the red color molecule, is a 12 pass transmembrane protein. It is not a canonical 7 pass transmembrane protein. It has very nice, it has very different structure where there is extracellular terms where the Hedgehog ligand goes and binds, and cholesterol also plays some role. We will not get into details of cholesterol, but remember patched is the Hedgehog receptor, remember, not smoothen, patched. So, whenever pathway is inactive, your patched is present in the cilium, your GPR161, which is another orphan receptor, that is also present in the cilium. And what these guys do, they make sure the pathway transducer, which is smoothen, does not localize to the cilium. So, smoothen is kept away from the cilium. When pathway is shut, when pathway is off, smoothen will not localize to the cilium. That is part A of the pathway. Is it clear so far? Teja, are you with me? No. Where are you? Where did I miss you? Sorry? That is fine. Okay. So, now what I am trying to tell you is that, so you remember there are 3 receptors that at least you should remember: GPR161, patched, smoothen. Smoothen is pathway transducer, meaning whenever, if what do we understand by transducer means pathway is activated. So, this information somebody has to pass on to the downstream, that job is smoothen's job. If you do not have smoothen, problem hai. Is it clear? This much is smoothen is transducer. G alpha, so smoothen goes and binds to G alpha, which is G alpha I. You guys remember we talked about there is G alpha S, G alpha I, G alpha Q, G alpha T. So it decreases cyclic AMP. No, if smoothen is active means pathway is activated, but less cyclic AMP. It is opposite to our thinking. You remember phosphorylation, we always think that it is activatory, but it is not always activatory, right? Now what I was trying to tell you guys is that, remember, patched is the receptor for Hedgehog. So first let us see what happens when pathway is inactive. So your sole purpose, like what happened in Wind, right Pratibha, what do you remember Wind? The sole purpose, there was a whole machinery, APC, GSK3 beta, then axine, then casein kinase 1. What did they do? They went and phosphorylated whom? Beta catenin. Where? Satyaranjan, where was beta catenin phosphorylated? in the cytosol. Then what happened? What happened to degraded? नहीं, उसके पहले क्या हुआ? Phosphorylated beta catenin was recognized by beta TRCP, which is the ubiquitin ligase. This ubiquitin ligase, Vaishnav, ubiquitinated phosphorylated beta catenin, so that it becomes a substrate, get to the proteasome and you degrade it, so that beta catenin cannot go to the nucleus and transcribe the genes. Now, your whole effort is going on to regulate GLI transcription factors, because GLI are the transcription factors. They will go and bind to the DNA, GLI response elements, and then they will drive the transcription of the Hedgehog pathway genes, right? Are you still with me, Teja? Now is it clear? Now what you want to do? You want to regulate GLI. Okay? I do not know why nature has come up with such a complicated system, but to regulate this GLI, we have 3 receptors which are working. 2 of them, they are trying to inhibit the guy who is a who is the pathway transducer, smoothen. So smoothen activation is very important, so that GLI can get activated. So when pathway is off, your smoothen is off, so that GLI can become off. Is it clear now? Little bit. Harshita, did you understand what I said? Good. So now just be with me. We saw who is the receptor for Hedgehog ligand? Patch. When pathway is off, there is no ligand. Patch is very happily sitting at the cilium. Okay? GPR161 orphan receptor, but negative regulator, again sitting with the along with the patch sitting at the cilium. What these guys do? And then your smoothen is kept away from the cilium. That is part 1 of the off state. You guys are with me still, right? What happens? Your GLI transcription factors. Now in GLI also there is GLI1, GLI2 and GLI3. Okay? I am not making it up, it is there in the book, so we have to understand. So there are 3 GLIs. And out of these 3 GLIs, GLI1 when we talk about GLI1 is like beta catenin, either it is there or not there, meaning when pathway is shut down, it will be degraded, when pathway is activated, it will be made, that is GLI1. GLI2 and GLI3 are very interesting. When pathway is shut down or when pathway is off, this GLI2 and GLI3 gets processed or partially cleaved, making them the GLI repressor forms. So in case of Bint pathway, it was very easy. You degrade beta catenin and don't worry about it, right? In case of Hedgehog signaling, what is happening? Your GLI2 and GLI3. So if you, what happens with that? Your GLI transcription factor gets partially degraded or partially processed, so that it forms a repressor form. Did you guys follow what I am saying? In case of Hedgehog signaling, remember, pathway is off, GLI will be repressor form. Pathway is active, GLI is active form. Simple. How does this GLI repressor form happen, Sudha, is what we will see now. We saw PAST is present, Hedgehog, GPR161 present in the cilium, smoothen is away, Now what happens? Your GLI transcription factors, they cycle through the cilium. They are taken to the cilium, they go to the tip of the cilium, comes out of the cilium. That is what is happening continuously. Who is very important to take these molecules into the cilium? There is a protein called KIP7. It is a kinase molecule. Okay? And when pathway is inactive, Aman, just be with me for some more time. When pathway is inactive, there is one more protein called SUFU. It is called as suppressor of fused. Fused is nothing but the glee in Drosophila. So, suppressor of fused means this guy is suppressing your glee transcription factor. What SUFU does? It goes and binds to your glee transcription factors. What we are talking is when pathway is inactive, Rutuja, pathway is inactive, everybody is trying to make sure that glee is in repressed form. How it is happening? There is an adapter protein kind called SUFU which goes and binds to glee. So, what happens? Your KIP7 is not able to take it and make it, right. So, SUFU when it is bind to glee this transcription factors, your protein kinase A. Now, there is a cascade of phosphorylation which is going to happen. Now, you guys will have to be little bit with me before we talk about what kind of phosphorylation happens. You have glee transcription factor. You guys know pKa. What is pKa? Very good. Who activates pKa, Tanvi? Cyclic AMP. Now you can understand why GPR161 is G alpha S coupled. GPR161 is G alpha S coupled because it can make more cyclic AMP, activate more pKa will go and phosphorylate glee. Right? When glee gets phosphorylated by pKa, that is the first phosphorylation. Then who comes? So, pKa, GSK3 beta, casein kinase 1. These guys will always be there. Even in your Wnt pathway, these two guys were there, no? GSK3 beta and casein kinase 1. Here there is additional guy pKa. Who is activating pKa? GPR161. Because more cyclic AMP, more pKa activation, more phosphorylation of glee. So far we are going good. Once glee gets phosphorylated, again same ubiquitin ligase which was doing the job in Wnt signaling comes here. Beta TRCP. It will come and ubiquitinate your glee which is over here. Is it fine? And nobody phosphorylates GSK3 beta. GSK3 beta is a kinase. So, GSK3 beta will go and phosphorylate glee. Everybody phosphorylates glee. Yes, Gaurav. This is glee. So, this we are talking about glee 2 and glee 3. glee 1 का क्या फंडा है? It's similar like GSK beta catenin. वो उसको पूरा डिग्रेड कर देगा. glee 1 का मत सोचो, just glee 3 का सोचो right now. Glee. Yes. Maybe it does for glee 1, but glee 1 completely gets degraded. In this case, we are talking about partial degradation. Glee 1 doesn't have repressor form. Only glee 2 and glee 3 has repressor form. Shine, are you okay?
Sir, this is all happening in the
Cytoplasm.
cytoplasm when the pathway switch off. Then only
Yeah, when pathway is off.
but when pathway is off, then how it is like forming pKa more?
So that's the beauty of this pathway. That's one we do not know what is the ligand for GPR161 because GPR161 is the molecule which is making more cyclic AMP. Very recently, if I am not correct, they have shown cholesterol has some role into that. So now that you are asking me, don't blame me if I make it very complicated. So there is outer leaflet and inner leaflet of these membranes, right? So there's cholesterol concentration present in the inner leaflet. So this patched has some hollow tube like region where your cholesterol can go and sit into, okay, and people have shown, and in fact this cholesterol is the one which might be activating your GPR161 indirectly when pathway is inactive. So the concentration of cholesterol in the lower leaflet, in the lower, upper, lower, whatever, outside, inside, whatever, this leaflet is the one which determines the activation. but I don't want to get into all that detail. There were I think there was one nature paper, one science paper or cell paper came at a time when they discovered there are two groups which keep talking about the structure of PAST and the role of this cholesterol binding domain in activation of the pathway. So, it is a debate which is there in the field. So, we do not want to get into the debate because I do not know much about it. But the point is, just remember that GPR161 is somehow getting activated and that is how it is making more cyclic AMP, which activates PK. PK will go and do the priming phosphorylation, then your GSK3 beta, casein kinase 1, they phosphorylate, and they phosphorylate this GLI, it becomes substrate for beta TRCP, which will come and ubiquitinate it. And this is your K48 linked polyubiquitination. Then what happens? This GLI becomes repressor form, meaning it gets partially cleaved by the proteasome. Now, it is again the mystery, how does the partial cleavage happening or partial cleavage processing happening, because once a protein enters a proteasome, it will completely get cleaved, but I do not know why there are so many fundamental questions still in this pathway. This pathway is known for over 50 years, no more than 50 years, but still there are so many questions how it is happening. Now, just take my word that it gets partially cleaved, kaam khatam, and then we have repressed GLI. This repressed GLI goes and binds to the GLI response element and somehow inhibits the transcription. That is the whole pathway Satyaranjan. Did you understand? Do you want to repeat it quickly, whatever we discussed? No, let us revise. He is like, I am fine.
The GPR161 is activated and cyclic AMP level increases so that the protein kinase A get activated and it will phosphorylate on GLI.
Right.
Subsequently, the GSK3 beta and the casein kinase 1 also phosphorylate on GLI, which became the substrate for ubiquitin ligase, which is beta TRCP.
Very good.
So that it will be polyubiquitinated and it undergo partial cleavage.
Very good. So, and remember this GLI is bound to whom? CuFu, right? So, CuFu is keeping it in cytoplasm. So, it is very important to keep it in repressed form. Vaishnav, are you following it? Yeah, so we will come to that part now. Is it fine? Rishika, did you follow what? Okay, good. Now, when the pathway gets activated, what happens? And this is another question, we do not know how it happens, but the GLI gets accumulated at the tip of the cilium. As you can see, I have shown it very nicely that it gets accumulated at the tip of the cilium. And who is very important? KIP7 is very important to make it accumulated at the tip of the cilium. And somehow, at the tip of the cilium, your CuFu gets separated from GLI. So, there are two events which happen before that, Teja. First thing, when pathway is activated, meaning your Hedgehog ligand is going and binding to whom? Vaishnav, who is the receptor for Hedgehog ligand? Patched. So, Hedgehog goes and binds to the patched. Once it binds to patched exits the cilium. Patched is no more present in the cilium. When Hedgehog is activated, GPR161 also exits the cilium. What is happening? Which means that cyclic AMP will be less. To make it more less, you have smoothen in the cilium, because smoothen is G alpha I coupled, which means that there is very less cyclic AMP, which means that the chances of GLI getting phosphorylated are less, and then GLI gets accumulated at the tip of the cilium. Some mechanism happens, and then your GLI gets separated from CuFu, and then it gets activated form. And this GLI activated then goes to the nucleus and drives the expression of Hedgehog pathway genes.
How exactly is the repressor GLI and the activator GLI
Are different? Yeah. How?
How are they different, like on a molecular level, how are
Hmm.
they different, the repressor form and the activated
So
form?
glee phosphorylation happens at the C terminus. That's when the phosphorylation motifs are there, and then it gets cleaved. So if you have, so full length glee will be present. I mean, I can pull out one of.
So the same molecule acts as a repressor as well as
Yeah, its repressor means it's not going to do active transcription. It just goes and sits on the same side, but it doesn't allow.
stop the formation of a transcription.
Correct.
But the activated form will allow.
Activated form will allow. I'll show you how does it look. Let me see if I have this. Here. So you see this plus SAG. SAG is the pathway activator. Okay, this is the data from our lab, but you can see this will be helpful for you guys to remember how glee gets. So when your pathway is inactivated, this SAG is the pathway activator. Don't worry about that. Just remember this is something which is activating the pathway. When pathway is not activated, you see there is this band which is present at 75 kilodalton, right? That is your glee repressed form. It is not a full length form. The moment you add SAG, now you see there is one band which appears over here, which is above around 180 kilodalton. That is your glee full length which is active. And then there is tiny amount of glee repressor form as well. So this is how the glee processing happens. If you have 180 kilodalton. Yeah, but here it is because it depends how long you are activating the pathway also, right? So this one was done for 16 hours, but if you give for 48, 24, 48 hours, the repressor form will go down. And even if you have this tiny amount of glee full length form, it is sufficient enough to drive the expression of the glee target genes. So, and here, no, here we are just activating the pathway. Yeah. 60 hours we are activating the pathway.
So in that case, the desensitization mechanism, how
So here these ones is the way we are activating the pathway is taking care of the desensitization as well. So there are multiple ways of activating this pathway. What we are using, this SAG is nothing but a smoothen agonist. It's going and activating smoothen. But if you use the natural ligand, it will have different course of activation. So that's how it is. But what I wanted to show you guys is this particular thing where how the repressor form versus the full length form is different. Full length will be full length protein, repressor form is cleaved, which is almost half of the size of the, not half, like around 70, yeah, 30-40% of the full length form it will be there. Oh, that is something that we are trying to study in my lab. There is a molecule.
So I'm confused about the fact that glee has three forms, 1, 2 and 3. So 2 and 3 form is repressor form, but 1 does not.
Glee 1 is like your beta catenin, like it doesn't have repressor form, it will get completely degraded.
So at the time that this process is going, maybe in the off state, so all the 3 forms are present. Right?
Off state, all the 3 forms will be present in a very minimal state, or even if they are present, they will be in repressed form.
So 2 and 3 will be in the
Repressed form
and 1
will be very less.
So when it's on, then 2 and 3 will be in the full length form and 1 will be present properly.
Correct. And sorry, go ahead.
2 and 3 all sits under same gene, target gene.
The thing is, the glee 2, glee 3 and glee 1, they all have same target genes, but sometimes what happens, your glee 1 comes first, initiates the transcription, and then glee 3 takes over. It is something like what happens in your DNA replication. DNA polymerase alpha is the one which comes first, initiates the primase because it has the only enzyme which has primase activity. It will make, now Zoya can remember what we taught. So it will make the primer, and then the elongation will be taken by another enzyme. Similarly, your G alpha, glee 1 can come first, activates the pathway, initiates it, and then subsequently glee 3 can take over. But again there is the context, there is a tissue dependent context to this whole pathway. It is not that every tissue same process is happening. In gonads it might be different, you know, or in your what brain, during the neural tube development another gli might come. So, gli3 might be dominant at some place, gli2 might be dominant at other place. So, there is some tissue dependent context to it. It is more complicated than that. So, just remember that there is tissue dependent expression and activation of these molecules would happen, and gli1 is something which might be at basal level. It is not some places it might not drive the transcription as good as gli3, then gli3 becomes important. Gli activator is activated meaning it separates from CFU. So, when it separates from CFU and it is present at the tip of the cilium, so it is not getting phosphorylated anymore, because you remember GPR161 removed out of the cilium, smoothen is at the cilium, which means cyclic AMP is very less cyclic AMP, less PK activation, then there is less chance of gli getting phosphorylated, and then your CFU gets separated. We do not know how CFU and gli get separated at the tip of the cilium, but what we only know is that they get separated. The mechanism, then it is separated means it does not get repressed anymore, means it will not have partial cleavage happening. So, it is full length activator form.
But in the case of gli repressor, there are too many processes by formation of gli repressor, like phosphorylated, then
Yes.
But in the case of like activator,
There is no need not to have it, because the only thing is here activator in the sense it does not get cleaved. You have full length protein in that sense. Anything else? Did you guys understand what we discussed? It is little bit complicated, there are too many molecules, but I think Teja will manage, right? Good. Do we have questions?
like it is in activated form. The example which you gave, like they eat those plants. In that case, how that hydroxyl
Cyclopamine goes and inhibits the whole pathway. So essentially gli will be always repressed, so gli transcription factors will not go and bind to the gli response element. So the downstream of gli is what? Downstream of gli is snail, you know, the gli transcription factor itself. So those are the very important. Snail is another transcription factor which is important to go and do another job.
So like this patch one was like after on-state it is going outside of the cell.
Out of the cilia. No, it will not do. In case of vertebrates, it will not do.
So in the on-state, the GPR161 and patched are not in the cilium, just moved there.
Yes, that's how the whole pathway is regulated.
The opposite of what happens in the
Off state, correct.
And how is this gene transcription factor, how is it moving into the nucleus and in the normal
Yeah, it's a regular process. Yeah, there is nothing different which happens. I mean, if you guys are interested, you can go back and check some of the papers which we have published, and we have really shown how GPR161 comes out of the cilium. That was what my postdoc work was, to identify the mechanism, how these receptors are recognized and they are removed out of the cilium. So there was a Journal of Cell Biology paper published in 2020 November, and the developmental cell published in 2023. You guys can access, then you will understand the mechanism what we proposed. So this essentially these GPCRs will be recognized and removed out of the cilium, so that only upon activation they are removed, not when they are inactive. So, and this is by now it's very well established. I'm not only me, there are other people who also publish, who also have shown that these smoothen is very well given. So when pathway is inactive, we see very teeny tiny amount of smoothen in the cilium, but the moment you activate the pathway, goes up. Do you have any question, Tanvi? No question? Pratibha, did you understand everything? Yes? Good. What about you, Shayan? Okay. Perfect. Thank you very much. If you don't have any more questions today.
Sir, like how it will get deactivated, like completely on-state?
No, there is another mechanism. Then whenever pathway gets activated, I think the inactivation happens, there is some kinase which goes and phosphorylates smoothen, and then smoothen gets inactivated. If smoothen gets inactivated, the pathway will get inactivated. Smoothen is the key molecule here. Smoothen knockout mice has very interesting phenotype. It is pretty bad.
Sir, are you trying to find out how these sulfurs get separated from?
No, I am not doing that because there are other labs who are doing it. So this we are not working on understanding these molecules. We have some other molecules to look into how they regulate the pathway. We showed. So what we do there was so I can show you how GLI1 looks versus how GLI3 looks. So you can see very clearly, right, when pathway is shut down in minus SAC condition, you can see very bare minimum amount of GLI1 levels are there. This is also around 150 kilodalton protein. But when pathway is activated, you can see very nicely. You have to compare this lane versus this lane, right? And you can see it is stabilized. So whatever treatment you give. So the way we do this experiment is that you take the cells, you serum starve the cells, and at the same time you add your Hedgehog ligand or smoothen activator SAG to it, so that cilium. So serum starvation we do because you want to form the cilium. So cilia is formed when cells exit the cell cycle. So in 2D cell culture system, we serum starve the cells. When you serum starve the cells, cells get arrested at G0, G1 phase. In G0 phase, there is the cilium which is very nicely formed. So I can show you how does it look. You can see this one. Do you see this tiny mark over here? That is the primary cilia that we marked, and this is your GPR161. You see this yellow color receptor? That's what we have marked. It has a neon green tag onto that, so you can image how GPR161 looks like. That's how it looks like into the cilium. It is just small tiny rod like this. So coming back to this image, अरे कहाँ गया? हाँ. And then what you do is you activate the pathway for 16 hours, and then you collect the cells, lyse them, and then run a simple Western blot and detect with GLI antibodies. We are not doing anything different in this. I mean, we have this particular data has a lot of paradigms that we have tried to use, but that's the way you activate, you seed the cells, serum starve them, at the same time you activate the pathway so that cilia is formed. When cilia are formed, it will go and activate those receptors.